So after studying this module, you shall be able to learn about the various areas within the occipital lobe, learn the functions of occipital lobe, learn about major deficits related to the damage of various areas in occipital lobe, learn about various kinds of agnosias, learn and understand disorders of integrative function in vision, and learn about the disorders of neglect. At the extreme posterior part of the cerebral cortex, there is a small region called the occipital cortex. It is the smallest lobe or area of the cortex which occupies only 12% of the total surface of the brain. Damage to this area produces blindness, which may be partial or total depending on the extent of damage. The occipital cortex may be divided into primary, secondary and the associative areas. Brodmann area 17 is the primary visual cortex, area 18 is the secondary, and the rest of the occipital cortex is associative. The primary visual area is connected to the lateral geniculate nucleus or LGN of the hypothalamus, from where the visual information is projected onto it. It provides the basic sensory experience of the visual sensation. The visual information is instantly transferred to the secondary area, where the interpretation of the given sensation takes place. The message is then relayed to the associative areas. There is elaborate projection of information to this area from other lobes of the brain, especially the parietal and the temporal cortices. The visuospatial perception and the integration of the auditory and the visual information is thus possible due to the function of the associative areas of the occipital cortex. The perception of the vision becomes highly cognitive due to the integrative mechanisms of the associative areas. The primary visual cortex, that is the Brodmann area 17, is known as the V1 or the visual one. It is located within the calcarine sulcus. It is also called the striate cortex because of the presence of stripes of myelin sheath when viewed under the microscope. Another name for that is the stria of Jennari. Areas outside the V1 are known as the extrastriated regions. They are specialized to interpret input related to motion perception, perception of static objects, color perception, complex pattern recognition and visuospatial information. Let us now understand the areas of the occipital lobe. Brodmann area 17 by V1 by primary visual cortex is located on the post-posterior end of the occipital cortex. It seems to have developed first in the evolutionary cycle of the development of the occipital cortex. The primary visual cortex may be further divided into 6 distinct layers of cells. This constitutes a thick mass of grey matter in the cortical region. The 4th layer receives information from the lateral geniculate nucleus of the thalamus. Various segments of the 4th layer receive input from the magnocellular segment of the LGN and the parvocellular pathways. There is a precise topographical representation of the space on the V1 of the visual cortex via cells on the retina. So, it is also known as the retinotropic mapping of the visual cortex onto the primary visual cortex. It was essential to have developed for the accurate spatial perception and location. A total map of the spatial information outside gets depicted on the visual cortex area V1. If an imaginary central line is drawn in the visual field, it may be mentioned that the lower half of the spatial world outside is represented on the upper back of the calcarine fissures, whereas the upper half of the visual field is focused onto the lower back of the calcarine fissure. Mapping of the outside world onto the visual cortex also includes the blind spot and the fovea. The blind spot is a small region which does not allow any visual sensation to take place. It is found in all primates. The importance and the evolution of that is still not well understood. The fovea is considered to be the point of the clearest vision. It is located at the central line of the vision. It is described as the center of cortical magnification since a large part of the spatial world is mapped onto this small region. However, it is a very thick mass of cells which can process large amounts of visual information. The neurons of the primary visual cortex or area V1 get instantly activated to discriminate sharply between features of a complex visual input as soon as the stimulation reaches here. This activation remains for about 40 milliseconds. Small variations in colors, spatial orientations and visual localizations can be responded to or perceived by these cells. Such cells also possess the property of supporting ocular dominance, which is a special feature of all organisms having binocular vision. The ocular dominance refers to the domination of one eye over the other for processing visual information. Such cells also possess the property of supporting ocular dominance, which is a special feature of all organisms having binocular vision. The ocular dominance refers to the domination of one eye over the other for processing of visual information. Such dominance is maintained at the level of individual neurons in the primary occipital cortex. Another special feature of the V1 cells is that those having similar properties such as responding to the left eye, etc., get together to organize themselves in the cognitive columns. They facilitate the task of differential perception of directions, locations and orientations in the spatial world. They can also analyze the temporal aspects of the stimuli and thus contribute to the perception of movement of the visual stimuli. Analysis of speed of movement is also a part of this perception. An integration of temporal aspects with the perception of space is crucial to this perception. After about 100 milliseconds, the neurons of the V1 area become attuned to the global perspectives of a visual pattern also. It happens due to the activation of the higher cortical areas and the projections from the pyramidal neurons. The diagram highlights these areas. The feed forward as well as the feedback systems are operative with these neurons. The feedback systems exercise modular effects. It suggests that the feedback coming from the higher cortical areas of the occipital cortex modify the responses of the V1 neurons. The modification takes place with the integration of the contextual fields into perception. The investigators have revealed that V1 neurons pick up information about the contours or the visual contrast points from the stimulus array. A line dividing black and white may be primarily picked up by these neurons. It is only when information is projected onto the nearby areas of the visual cortex that the full brightness of the scene is captured. The most obvious local contrast points are primarily captured by the neurons of the V1 area. The association areas of the occipital cortex perform the most intricate function of stimulus integration, pattern recognition and visuospatial coordination. The association cortex has several specific regions such as V2 to V6. They are collectively known as the Brodmann area 19. Visual area 2, V2, is also called as the pre-striate cortex. It is the first area in the associative cortex. It has fiber connections with V1 directly as well as through pulvinar, an important area on pons in the form of the feedback system. It is also connected to other areas of the association cortex. Neurons of the V2 have many functions similar to those of V1. They process input of spatial orientation, color and shape, etc. They respond to the presence of sharp contours and recognize items as figure or the background in the visual field. In addition to that, V2 cells are modulated to focus attention on an object. They are involved in the processing of slightly more complex patterns. They show activation also when an individual comprehends multiple orientations within a single small visual field. Such neurons play an important role in visual memory. It has widespread connections with the hippocampus, which is essential for the storage of the object recognition memory. If this area is damaged, the memory related to the recognition of objects is badly affected. The area V2 is highly interconnected with other areas of the visual association cortices. In front of the V2 there is V3 located in the upper and the lower parts of the cortical region. They process much more complicated input. For example, different neurons respond to different colors. They also refer to various combinations of the visual stimulus such as color combined with size or movement, etc. Area V3 has been associated with the processing of objects in motion. Such areas have representation of the whole visual field in a topographical manner. Thus, perception of the movement of large objects as a whole is provided by these areas. They receive projection from the parietal and the temporal cortices and are involved with the task of integration of information of all of these three lobes. Area V4 is located and is the largest area in the associative cortex. Its location is anterior to V2 and posterior to an area called the posterior inferior temporal area. Like other regions, it is also highly connected with other major regions of the associative cortex. The major function of this area is to modulate the function of attention. The tasks related to selective attention cause strong neuronal firing in this area. Area V4 contains all the properties of V1, V2 and V3. Additionally, it is attuned for more complex pattern recognition such as that of geometric shapes. Besides that, this area plays a significant role in the recognition of various colors and forms or shapes. It recognizes the salient features of a stimulus array and is especially attuned to receive messages from the straight line of vision. The spatial features of this area indicate changes according to selectivity of task to be performed. The cells of the area V4 show plasticity, that is, they adapt to the changing situations in the visual field. Area V5 has synaptic connections with some cortical as well as subcortical regions of the brain. It receives input from other visual areas of the association cortex, lateral geniculate nucleus of hypothalamus and the pulvinar region of the brain. The V5 controls movement perception in a global manner. The local motion is integrated with the overall movement present in the visual scene. The area also controls eye movements. It has also been demonstrated that the cells in the V5 respond to the input according to the direction from which it is projecting onto it. The projections of this area also reach the frontal eye field of the frontal cortex and the lateral intraparietal area of the parietal cortex. This area receives information directly from V1 and also from other associative areas. Area V5 is also known as the middle temporal area or MT. It integrates information between occipital and the temporal cortex. Several electrophysiological studies have been carried out to study the functions of MT. A large number of functions related to the perception of movement, coordination of eye movements with the moving stimuli, perception of temporal aspects, that is, the speed of moving objects, etc., are similar to those of V1, but they are carried out at a more complex level here. The MT area is also attuned to the vertical or the horizontal plane of the moving stimuli. Specific stimulation of certain neurons in a monkey has been found to produce upward or downward movement as revealed by the electrophysiological studies. Such neurons respond to the direction of the movement as well. Area V6 performs higher order task of integrating vision with other sensory modalities. Well students, now let us understand some of the disorders of the occipital lobe. The primary function of the occipital lobe is to provide visual sensation to the organism. Total damage to this lobe in both hemispheres lead to total blindness. Damage to this region in only one of the two hemispheres may cause partial blindness with other distortions of visions. Lesions in the occipital cortex may produce illusions and other distorted perceptions such as the objects looking smaller or larger than what they are, objects having abnormal hues or objects totally devoid of color while they actually may be quite colorful. The lesions may also produce hallucinations, that is, seeing objects without the presence of any visual stimuli in the environment. Lesions in the associative areas are more critical. For example, lesions in the temporal parietal occipital areas may cause impairments in writing as well as comprehending words. The latter is known as the word blindness. Let us now understand apperceptive and associative agnosia. Not being able to recognize objects at all or to get confused with objects seen in a different light, color or orientation is called as agnosia. Agnosia literally means absence of knowing, and it can happen with any sensory experience. Agnosia is a failure to recognize objects in their totality. Perception of a visual object, for example a rose, involves integration of its shape, color, texture and smell. A person with agnosia will recognize each of these features independently but will not be able to integrate them all to have the perception of a rose. If these sensations are independently added to the experiences, they may be integrated. If he is simultaneously asked to smell the flower, touch it and look at it from different angles, he may be able to recognize the flower. Thus, integration within the visual modality may be lacking even though a cognitive addition could be done. Such agnosias are always modality specific. Apperceptive or associative visual agnosias can be differentiated on several criteria. In apperceptive agnosia, the patient cannot synthesize the information coming from various aspects of the scene such as shape, lines, colors and size into the global picture of the percept. Their perception is in small units. They may name various bits of the percept but may not be able to see it as a whole. On the other hand, patients suffering from associative agnosia find it difficult to assign a meaning to the visual image that they perceive at the moment. Such patients are able to synthesize information of the visual percept coming from its various attributes. For example, if they are shown scissors, they will pick up the scissors from the array of objects shown to them, but they will not be able to say what it is and how it can be used. However, the two kinds of agnosias generally occur together since they are caused by widespread damage to the cortical areas. The specific sites of damage for each kind of these agnosias get blurred because of overlapping areas. Now let us understand some other variations of agnosia. First is the visual object agnosia. Repeat, it is caused by the lesion in the right occipitoparietal region. Visual object agnosia involves the loss of recognition and comprehension of the meaning of an object even though the perception of the object may be intact. At times, lesions in the left hemisphere are also present. Several tests have been devised to screen out such agnosias. Prominent among them are Object Assembly Test of the Wechsler Adult Intelligence Scale, Benton Visual Retention Test, Reading Fragmented Letters Test and common test involving recognizing meaningful shapes in an array of ambiguous pictures. Now let us come to the next one, which is prosopagnosia. It is characterized by the impairment in recognizing faces that one is familiar with. It accompanies occipitoparietal and occipitotemporal regions in the right hemisphere. Matching of the facial features and recognition of familiar but non-relative faces are the tests generally used to diagnose this kind of agnosia. Coming on to the next one is color agnosia. This agnosia relates to the perception of color. Naming colors and differentiating between them is the primary impairment in this area. It may occur with aphasia or alexia. The major cause of this disorder is a lesion on the occipital or occipitotemporal regions in the left hemisphere. Some of the major screening tests involve color sorting by hues, intensity or by number of colors shown in the picture. Matching color names to color shades and vice versa is another test utilized for this purpose. In some ingenious tests, color of the objects inappropriately marked has been utilized to screen for this agnosia. Coming on to the next one is simultaneous agnosia. This kind of agnosia involves impairment in perceiving more than one object at a time. It happens when lesions appear bilaterally in occipitoparietal regions. They may occur in combination with inability to coordinate movements of the two eyes. The best screening method is to ask the patient to draw lines across the contours of various shapes. It is done with the eyes open as well as when the eyes are closed. It involves tremendous coordination of the movements as well as absorbing the shape characteristics. Coming on to the next one is metamorphosia. It has been generally established that lesions in the occipital cortex produces milder deficits, whereas those in the occipitoparietal cortex are more severe in their disabling effects. Lesions in the occipitotemporal cortex produce the worst kind of impairments. Metamorphosia is a severe deficit in which the perception gets distorted. Some of the examples are teleposia, in which even the nearby objects seem to be far off and very small in size, and pelopsia, where the objects seem too near and too large. In the case of lesions in the right hemisphere in the occipitoparietal region, pelanopsia may occur, in which the illusory effects superimposed on the objects may occur. The best tests for the diagnosis of such conditions are recognition of forms, moving stimuli and rate of critical flicker frequency analysis. Now let us understand the disorders of integrative function in vision. In day-to-day life, integration of visual sensation with the motor movement is extremely important. Due to damage or lesion in the cortical visual regions, some specific disorders have been identified. They may be described as follows. Disorders with visual spatial basis of movement. Lesions in the inferior parietal occipital region in the right hemisphere leads to this disorder of lack of integration between vision and movement. The tests are to make a patient reproduce the drawings made by the clinician while sitting beside him and also while sitting in front of him. Disorders with kinesthetic basis of movement or afferent apraxia. The lesions in this case are on the post-central gyrus on the opposite side of the imparted movement. It produces diffuse uncoordinated movements. In order to test this, the blindfolded patient is asked to place his hand on one position and then place the other hand at the same place in the same orientation. He may be also asked to reproduce the position of the hand as made by the clinician. Next comes the disorders with motor integration or dynamic basis of movement or afferent apraxia. Inability to continue a kind of movement and repeating the same movements again and again are the symptoms of this disorder. They occur due to the damage of the premotor area of the left brain. Specific tests developed for the purpose involves drawing of different designs, alternate patterns of tapping to be replaced, ring fist test and other requiring coordination of both hands. Next comes word fluency. Speaking or writing words fluently is an example of sensory motor coordination. The ability of motor behavior as well as of producing language resides in the frontal cortices. Thus, it is called a disorder of the frontal lobe. The test to diagnose it involves speaking or writing as many words as one can, starting with a particular given letter for which a particular time limit is allowed. Severe memory deficits. Memory deficits related to visual images is a disorder common with memory of other sensory modality images. It is associated with the damage of the temporal cortices and the areas on the hippocampus. To diagnose the problem of several memory tests are used. For example, paired associate memory test, memory of hidden objects, reproduction of stories and the recall tests involving retroactive inhibition tasks. Now let us understand what do we mean by disorders of neglect. This disorder is known as unilateral neglect, hemi-neglect, contralesional neglect, hemispatial agnosia, visuospatial agnosia or hemispatial neglect also. In this disorder, the limbs on one side of the body, commonly on the left side, hang as if they have no life. On close neurological examination, they seem to experience pain and can be moved by the patient, showing that their sensory and motor system are functioning normally. But the patient is not consciously aware of their presence. Some kind of brain damage is to be expected. The research indicates that the damage to the right parieto-occipital or inferior parietal area is the site of this damage. Well students, now let us summarize is that the occipital lobe encompasses the posterior portion of the human cerebral cortex and is primarily responsible for vision. Primary visual area, Brodmann area 17 or V1, is involved with the processing of visual information, its spatial characteristics and pattern recognition. There are 6 visual areas on the occipital cortex. Occipital lobe lesions cause seizures, which include symptoms like visual hallucinations, visual field defects, homonymous field defects, palinopsia and sensation of eye movements, eye pain, involuntary eye movement, nystagmus, eye jerking and eyelid fluttering. Disorders of lesions in area 17 include homonymous hemianopsia of contralateral visual field, eblospias, central scotomas and muscular sparing. Apperceptive and associative agnosias have been diagnosed as major occipital lobe syndromes. Other variations are prosopagnosia, color agnosia and simultaneous agnosia. Disorders with motor integration, efferent apraxia and disorders with unilateral spatial neglect, also known as contralesional neglect, hemi-neglect, visuospatial neglect are other forms of agnosia. Most of the disorders are associated with lesions in the right inferior parieto-occipital lobe or generally in the posterior regions of the cortex.